CSIR-IGIB Delhi. Sir will be conducting a lecture on genomic technology under the guidance of Dr. Bhavana Prasher Madam, a senior principal scientist at CSIR-IGIB, New Delhi. Sir, I request you to please proceed with the topic.
Thank you. I hope I'm audible.
Yes, you're audible, Vikas. You can introduce yourself and also your briefly tell about your PhD topic.
Yes, ma'am. So, good evening, everybody. So today I'm going to give a small lecture on the genomic technologies that are being developed and used today. So my name is Vikas MH, and I'm a PhD student under Dr. Rakesh Sharma. So our lab basically is a microbiology lab that deals with a lot of molecular biology stuff. What we basically focus on is metagenomics, using NGS technologies to study the microbiomes present in different environments. So my PhD topic is basically on the gut microbiome. So we have a project in our institute called the Phenome India project, which a lot of PIs are a part of. It is a collaborative project which is being conducted all over CSIR institutes across India. And as part of that, we are doing the gut microbiome analysis for all these participants. So I'll be telling a little more on this in the coming in some slides in the coming this thing. So can I start with the lecture?
Yes,
Yes, sir.
please start.
Yeah. So to start with, is that so why do we need to do DNA sequencing in the first place? So I think from your previous lectures, you people must be now familiar with the central dogma of biology, that DNA is the genetic material that has all the information that is being expressed in the cells through transcription through RNA, and then the proteins are the phenotypes that we see in all these organisms, in the living organisms. So basically we want to understand what these genetic instructions are containing in all the living cells, because if we can decode the fact that how this particular code is being translated into a physical entity, so we will be able to if we can control this particular process and understand, we'll be able to study the genetic variations and the mutations that are happening in diseases and diagnose them early and find a curable treatment for these things. This particular mapping is done by sequencing DNA. So DNA is basically just a simple sequence that is made up of 4 different bases called ATCG, purines and pyrimidines. Uh, so the early sequencing methods had 2 different methods where they were independently developed at the same time through chemical cleavage method and enzymatic method. So the chemical cleavage method was developed in such a way that say if you have a fragment of DNA and you are going to carry out 4 different reactions at the same time, and each one will be giving a different reaction. So say one of these chemical reactions gives a positive for one particular base, assigned for one particular base. So you decide that, okay, the base for this particular base at this particular length. So like this, you need to keep doing it again and again, again and again. So say for about 500 nucleotides, you need to do the reaction to 500 times. So into four, you can imagine like that's a very tedious process. So then came the so simultaneously another method was um, developed by Sanger, which involved enzyme. So these were the first people Maxim and Gilbert. They developed the chemical method, and the Frederick Sanger developed the enzymatic method of Sanger sequencing, back in 1977. So the Sanger sequencing method is a very clever way of trying to decide the sequence in a very long length of DNA fragment. So what they decided to do was that, say we introduce different so you must be familiar with replication now. So whenever the polymerase enzyme, it carries out the replication of a particular template DNA, so it will incorporate the corresponding complementary nucleotides into the strand. So here the idea was that, say if we introduce a defective nucleotide, so this is the normal DNTP. So say we introduce a ddNTP, so it's devoid of oxygen at this particular position, and when this particular thing is introduced, this will be able to get into the strand, but the reaction will not continue. So what happens is, whenever a particular base is added, that a defective base is added, the reaction stops there only. But there are also enough normal DNTPs which can allow the molecule to carry on the reaction as well. So whenever there is a at the beginning of the reaction, so we add all the four modified DNTPs and the normal DNTPs along with the primer and the enzyme with the template DNA. So when we do this, when the reaction proceeds, so whenever a defective nucleotide is being added, ddNTP, so at this time the chain extension stops, because the polymerase enzyme actually requires this OH group to continue the sequencing. So once it stops, what happens is that the fragment is stopped there itself. But the rest of the molecules where there might be incorporation of a normal DNTP, so the reaction continues. So once we do this enough times, we will be able to get a, a pattern like this. So when we separate the DNA fragments in our sample, we will be able to see that, okay, at T, so there is a fragment here, right here, that has traveled the farthest. So we know that T is there at this particular sequence and at this length. So now when I try to look for my DNA sequence, so it starts from here, right? So I know that the sequence is T, C, G, A, like this. So this is how it was done manually. So so later on, what happened is that they automated this process and set up a computer where it can detect these particular variations through laser diode, so that you don't have to do it manually. So once this was automated, the sequencing technology had a leap where one could process this particular 1,000 base pair, up to 1,000 base pairs of length of DNA in one go. So Sanger sequencing gave us the very basics of sequencing, where we started that sequencing a particular genome is very much possible. So since then we have come a long way, and the fact that Sanger method gave us that hope. So with that hope, we started sequencing larger fragments of DNA, such as the human mitochondrial DNA, which was sequenced in 1981, and then the launch of the Human Genome Project was in 1990. So so the project started in 1990, and it was carried out for the next 13 years, for up until 2003, when the first draft of Human Genome was released. And in 2005, Roche came up with a automated genome sequencer, where this particular task of which was done in around 13 years was possible in terms of days. So this particular jump gave the sequencing technologies a boost. And from here onwards, the Human Microbiome Project, which was launched in 2007, also gave a lot of opportunities where the NGS could be used in biological experiments, and at the same time the NGS technology started developing. So with the invent of nanopore sequencing devices and PacBio sequencers, where longer fragments of DNA were possible. So with the and these particular technologies gave us uh, uh, the chance to analyze very long lengths of DNA at the same time. And at today, we have completed the human genome project, human microbiome projects, and so and the NGS technology has been developing more than ever. So the sequencing technology has been going through a lot of changes, and the approaches for sequencing the DNA fragments is also changing. So the very first Sanger method used something called, so they required a primer. They will always need a primer to start the sequencing. So what you basically do is you take a fragment of DNA, say you know the first half of the sequence, so you make a primer for it, and then you synthesize the next half of the DNA up until where your polymerase enzyme can catalyze the reaction. So you know that, okay, from here 1,000 base pairs you were able to determine, and then you design the next primer, and then you do the next primer. So this was done. This was how the first human genome project was started. So but by the time around 2001 and 2002, what they realized was that this is a very tedious method, and some private fund, people from private industry also got involved to speed up the process. And what they came up with was the shotgun method, where you can just take the whole length of the DNA, and then you fragment it into pieces without having to know where you are cutting it. So you cut the DNA enough that you obtain fragments of less than around 600 700 base pairs, and you sequence all these fragments, and then try to find a consensus sequence after the sequencing is done by computational methods. And but this is a tedious process. One more method that you can use is the target gene, or the by targeting genes and regions. So say you want to just go check for the cancer gene, for breast cancer gene. So you just need to take out design primer for the BRCA2 gene, and then check for it. So and in case of bacterial sequences, like 16S is the sequence that is used to identify bacterial taxonomy. So you just need to have the primer for it, and then synthesize the gene, and by targeting that particular length only, you can determine whatever you want to get out from the experiment. So nowadays we mostly use the targeted gene approach and the shotgun approach, where shotgun approach is mainly used for a human genome and the microbial genome. So with the invent of these automated sequencing technologies, so the sequencing methods where they started to be named as next generation sequencing. You a workflow for the human genomics. So say you want to sequence your DNA. So what you can do is you can take the cells from your cheek or from blood, and then you try to extract the DNA these cells, and their QC is done to check the integrity of the DNA and the quality. So this whole process can take about 3 hours. And then there is a library preparation method, where the taken DNA so is fragmented into smaller pieces with shotgun method, and this fragmentation can be enzymatic, or it can be done using some shearing techniques, mechanical shearing techniques. And then you add adapters, where you get to identify which are the DNA from one particular sample. So you basically barcode these samples, and then all these together, they are called the DNA libraries. And then these are put into the sequencing machine. In detail, we'll see in the next slides. So they are put in the sequencing machine, and once the sequencing begins, the base calling is done. And this particular base calling will give you a lot of reads. So these reads are later either aligned to a reference sequence, or it will be de novo assembled. And this particular assembled sequence, which you can use it for aligning and identifying variants. And once you identify the variants, you will be able to know from the reference genome that is there any mutations like insertions, deletions, or is it in consensus with the population uh, variant that is present. So basically, this is the basics. This is the basic pipeline that is followed in any of the NGS um, workflows. So that will have the nucleic acid extraction and QC, and then the library preparation, and then sequencing, base calling, and then the bioinformatic analysis begins, where the data will be processed to check for the relevant information that comes from DNA. So all these feeds can be in today's time can be achieved from 3 different technologies which are offering us this particular applications. So the one that most people around the world use is Illumina. So it uses a sequencing method, something called sequencing by synthesis. So it uses the normal replication process that happens in a DNA uh, of a DNA molecule using polymerase enzyme, and we use that, and we leverage that fact to sequence our to actually determine which base is present in the sequence of DNA. So this can sequence up to 600 base pairs of length of DNA, and the highest throughput that you can get from these machines from it can be up to 6 TB per for one run. And so you can imagine the amount of data that comes from these runs is very high. And then comes the Oxford Nanopore. So these two technologies are different from the Illumina in the sense that they offer uh, us the opportunity to sequence large segments of DNA. So in case of Oxford Nanopore, it can go up to 2 MBs. So it all depends on how much quality DNA you are able to isolate. So based on that, you can sequence any length, higher lengths of DNA. And the flow cells that are there, so for this particular device, it can generate up to 50 GB of data per run. And the PacBio is also a long read sequencing technology where you can sequence up to 100 KB DNA fragments, and it can generate up to 500 GBs of data per run. So we'll look into each of these technologies in detail. So the Illumina technology, in the Illumina technology, the sequencing is done by sequencing by synthesis. So what do you mean by sequencing by synthesis? So synthesis is basically I am trying to synthesize a complementary DNA fragment by using the DNA polymerase enzyme. And in the process, I'm manipulating the process in such a way that I'm able to identify the identity of the uh, nucleotide present. So this is called sequencing by synthesis. So whenever DNA fragment, it is fragmented into smaller pieces, and then adapters are added so that the libraries can be used for the sequencing. So these particular adapters along with the barcode, so these adapters are designed in such a way that they have a complementary binding site on the flow cell of the machine. So when you have a flow cell, so these particular fragments, they will go and bind to these clusters. So once a particular DNA fragment binds to a cluster, it will be there is a step called bridge amplification, where one particular sequence it gets attached to the next one. So these are complementary sequences. So it multiplies in numbers just by uh, sequencing again and again. Multiplying again and again, uh, and this generates the clusters. So once the cluster generation is done, the sequencing process begins. So what we do here, in order to explain it in simple terms, so we sequence one base pair at a time. So one base pair is sequenced, say, so at that particular first base pair, so this particular cluster is having G as the base pair. So all of these will be generating a unanimous signal, right? So, and this one will be generating a signal for C. So all these are fluorescent labeled. So whenever the these are added, the fluorescent dye that is attached to these particular molecules is released. So when you look from the top, it looks like something like this. So each cluster is emitting a different color, which will be corresponding to one particular base. So at each reaction, so at each reaction, whenever it is going on, a camera captures the images of these reactions, and based on the intensity of different dyes, it determines which particular base pair came from which cluster. So based on this, we'll be able to identify the sequence in the reads, and all the reads that are sequenced in millions and billions of clusters will be aligned later to get a consensus sequence, which will be your original template. So the present day, the Illumina machines are one of the top of the class machines which are being used across the world, and the MySeq I100. So they have different ranges of machines which you can use, which you can use for different experiments. So MySeq basically gives you smaller data up to 30 GB, but what it can make up for is the length, where it can generate up to 600 base pairs of length. This can be used for smaller experiments where you need to try something before you actually go for it. And then NextSeq was um, developed recently, and this has the capability of sequencing up to 600 base pairs as well, and it can generate data up to 540 GB and about 1.8 billion reads in a single run. And these runs can the whole sequencing experiment will span. And the NovaSeq X series, which is the latest and the most advanced of all the machines that Illumina offers. So it can generate up to data of 16 TB in a single run. So and up to 52 billion reads. So you can imagine the amount of DNA, DNA data that can be produced by these machines if you can, um, generate that kind of stuff. And it takes up to 2 days to generate this kind of data. So here in all these machines, it depends on what kind of experiment you are doing, and based on that you can go for the flow cell and the machine that you want to use. So all these offer is that they can offer you a maximum read length of 600 base pairs, from 150, 75, 35 to around 600 base pairs, not longer than that. So nanopore sequencing technology is very much different from the Illumina sequencing in the fact that it is a physical process. So what they have here is that they have a membrane where protein is fixed. So this acts as a nanopore. So the DNA fragment is being passed through this spore, and each time a nucleotide is blocking the opening of the pore, it blocks a certain current that is being conducted across the membrane. So whenever there is no DNA, there will be a basal level of electrical conductivity across the membrane, and that will be recorded. And whenever a base passes through it, it blocks a certain area of the protein, and that electrical conductivity is transmitted. So each of the bases have very different, very distinct uh, the voltage drops which can be detected in real time, and this can be used to sequence the DNA. So like this, each of these pores are arranged into a flow cell where there are a lot of pores, way up to 2000 4000 pores in one particular flow cell, which can sequence up to 72 hours, uh, which can take up to MBs of DNA fragments if you have such longer So how does this work? So this particular nanopore that is being used in the process is nothing but a protein, uh, a protein nanopore, that is taken from the transmembrane of a mycobacteria. So it is called the MSPA. So this is the latest protein that nanopore technology is using, and the size and shape of this particular protein allows us to sequence the DNA. And nanopore is also coming up with different proteins which can sequence proteins as well. So basically, each amino acid will also have a distinct electrical conductivity, which can be measured across the membrane whenever it passes the nanopore. They are working on that as well. So when it comes to the nanopore machines, they are very much user friendly compared to Illumina. So Illumina will give you a lot of data, but the machines are sensitive and they need to be although these machines are also sensitive, but they are small, and you can take them anywhere. So when you see this is just like a pen drive. You can attach it to any laptop and you can start sequencing then and there. So the flongle adapter, which comes like this, a smaller one, it can sequence about GBs of data. It is used in very remote locations where one cannot transport the samples back in time. So what you do is you just take a good enough laptop with this particular device, and you can start sequencing on site then and there. And even min ion this particular adapter is for the flongle, but the min ion flow cell goes right into this device, and it can generate up to 48 GB of data hours typically. And if you want to generate longer high data for say some of your samples, you can use the promethion flow cells, which are not you cannot carry these are little bigger ones. So they can produce generate data up to 150 to 200 GBs, and these can be used for a higher level profiling. So PacBio is also one more technology that can sequence long reads of DNA. So here it also uses a similar method like how Illumina uses the fluorescent bases that are being added through and these are detected. So the same technology is the same process is also leveraged here to detect the base pairs, but the principle of the initiation of this particular process is a little different. So whenever you have a fragment of DNA, what they do is they add two end sequences of DNA called smart bell libraries, where these particular DNA make the whole template DNA into a circularized DNA. So once this starts, once the zipping starts, so this will be converted into a circular DNA. So the DNA polymerase, what it does is that it goes on replicating this particular strand of DNA inside this loop for multiple times. So this gives them the advantage that every time it sequences, all of these sequences are being recorded by this particular process, and you have enough consensus in the whole of the sequence once it is done, that you are able to get a very accurate base calling from this particular all of these reactions when they are accumulated. So how is this detected when say, but because in Illumina what happened is you have clusters, so you are able to do it, you are able to detect a single signal for each cluster separately. But how can one do this in case of PacBio as well? Because you are not restricting any clusters here. You are randomly adding all the smart bell sequences to all of the DNA sequence. So what they also have is that these smart bells are also designed to go and attach into small and very small chamber, which are called zero mode waveguides. So here they will go and attach where only one single molecule is permitted here. So once it is populating, the other sequences won't be able to populate this. So what this allows is that now you are isolating one particular sequence to one particular site. So now whenever you are able to detect the sequences, the fluorescence, it will be specific to this particular uh, chamber and the sequence. So that is how this particular these technologies we leverage the property of DNA to actually determine which base is present. And when it comes to the machines by PacBio, so there are different machines, different range of machines where there is a SQL. So this is a bit older model which can generate up to 30 GB data, and the newer models like Vega and PacBio, they can generate data up to 60 GB to 480 GB, and the length of the sequence can go up to 20 KB to 25 KB. And this gives a very distinct advantage for these sequencing technologies over Illumina, where the smaller reads are generated, and the runtime is almost similar here, from 12 hours to around it will take around 30 hours. So why timing is a factor that we look into is that when you are dealing with, say, some medical emergencies or medical treatment strategies where sequencing needs to be where the gene sequences of a patient need to be determined in a very small timing, where or else the patient might be in trouble. So in order to do that, so knowing these particular details also with designing these experiments. So when it comes to the applications of all these technologies, so at the genomic level, we'll be able to determine the single nucleotide variants, the insertion, deletion, copy numbers of a particular gene, the short tandem repeats. Um, I think most the talks lecture yesterday and day before yesterday, they have of these topics. And this, so when they are talking about that, this is how we detect deletions, insertions. So the actual process involved is this. That is how these fragments of DNA are uh, sequenced, and these are tedious processes where one has to isolate the DNA, make libraries and sequence. So when biology when biotechnologists actually use the word that we sequence something, so they mask the very effort. So it takes a lot of uh, analytical experiments that go into determining these sequences. So now that sequences have been properly automated, we can generate a lot of data in very small time. And when it comes to epigenome, the DNA modifications and the histone modifications that are done to the chromatin can also be detected. So when it comes to, say, nanopore technology also, so whenever a base pair, whenever a nucleotide is passing through with uh, you know modifications, so you get to know these particular modification, methyl modifications and all, by measuring the electrical conductivity. And the Illumina and PacBio are also into this, but they employ different methodologies. And one can also look into the transcriptome, where you can check what kind of RNAs are being transcribed, and which are further going into making the proteins. So because not all of the DNA is being transcribed into expression levels, most of the genes might be they can be dormant, or only a few of them are very say active. So in order to check that, you need to look at the RNA level. So but RNA is a very unstable molecule, which has a very small half life, and it can degrade with any a very small contamination as well. So how is the sequencing of this particular RNA molecules done? So the very study of this methodology is called the transcriptomics, where you will be able to sequence the RNA. So once you isolate the RNA from any of the samples, what you try to do is that you try to fragment them into smaller pieces and then convert them back to DNA. So this is called cDNA, and this can be achieved through reverse transcription. So we have RT-PCR techniques which have been optimized over a lot of experiments, and you can use these to make the cDNA, and these cDNA are then used for making DNA libraries, and then you can sequence them to get the map of this transcriptome. So now you have a sequence of the genome that you sequence from the genetic material, the DNA, and then you have a map of the transcripts as well. So now you know that which part of the DNA is being expressed, and how much of it is being expressed based on the copy numbers, like how much depth it has in proportion. So what are the applications of the transcriptomics? So the applications are, first one is gene expression, like I mentioned. The other thing is the alternative splicing. So alternative splicing is where a single gene is being transcribed into different fragments because of the during whatever modifications it goes through during the transcription, and during post transcription processes. So when this splicing occurs, so it is removing some of the introns and different combinations of exons as well, so which can give rise to different isoforms of the protein. So and these will have different activities, and they can give rise to different phenotypes. So knowing how this particular is, so just determining the genetic sequence does not support us in determining the outcome, because the process in between, the transcription, is being done differently at different times. So in order to determine this, transcriptomics is important. And the allelic gene allele specific expression, so where say we have different alleles of one particular gene from different chromosomes, and what is the level of their expression, because based on the how much expression they are, what is the proportion of their expression, the phenotype is being determined. uh, say in case of when we read about codominance, dominance, so those things we have seen that the alleles can have masked effect, or they can work together, or they can dominate. So in order to determine this, you need transcriptomics data and the modifications that are being done to the RNA. So uh, the modifications that so okay so the
Please share your slides again, Vikas.
okay, ma'am. So was I audible the whole time, or like did I miss
You are
any part?
audible. Just last, this modification part is where we lost you.
Okay, okay. So should
Yes.
I explain this again?
I think alternative splicing is over. You can start from phasing and allele specific
Yeah, okay,
expression.
okay. So in terms of when it comes to allele specific expressions, uh, different alleles of a particular gene are present. When they are present, they can have and when they are expressed in different levels, they can have a different effect. The combination of these alleles might be dominant, or in combination it can produce a different phenotype as well. So in order to know how these phenotypes are being determined, so if you know the expression levels of these different alleles, you can easily determine the outcome. And the post when there are modifications involved in terms of transcription as well, and most although we study most of the mRNAs which are being translated into proteins, there are a lot of RNAs which are not involved in any translation procedures, but rather they are uh, used they are used in signaling pathways, and these are called non-coding RNAs, and they have other functions depending on their modifications that are involved. So in order to by understanding these modifications also, we can predict their functions and how they are affecting the phenotypes. So the in so combining both the genomics and transcriptomics, the present day applications of genomics are uh, very wide. And here I'm just trying to show you the major applications of it, where we are able to make significant progress at the moment. So when it comes to diseases, cancer is one such disease where genetic information is being sequenced a lot to determine the genetic variants or the mutations that are happening in different cancers. And by determining this, we are trying to treat those particular cancers in with respect to those genetic variants. And the rare diseases, where the occurrence of the disease itself will be so low that you won't be getting too many cases that you will be able to properly study them at a population level and come up with a diagnosis and a treatment plan. And there are again infectious diseases where you go, like we saw during COVID, how infectious diseases can spread through, and how genomics helped us track these different variants when it comes to Ebola and other infectious diseases also. Tracking these strains, the virulent strains, and what gives them the ability to cause the disease, can also help us to come up with strategies for making vaccines and probable treatments. And microbiome. So microbiome sequencing microbiome can help us know what are the microorganisms that are present. And in case of human, it can be from skin, oral, or gut microbiome, where you'll be able to identify these organisms and how they are affecting our health. But not just that, microbiome can also be from different environmental locations as well. And so genomics has also been employed in pharmacogenomics, where how genetic composition of one particular person determines his response to treatment with one particular drug. So how are they responding based on their genetic composition? And sequencing also has been a very helpful tool. The NGS, especially the NGS, is that now we are able to carry out large scale population studies where we can track the different genes and different variants, how they are occurring and causing any changes in the phenotypes. And sequencing can also help us come up with new stuff. So say novel proteins like enzymes and antibiotics. So enzymes in the sense, so so I will give you an example from what my lab does, is that say in our lab, what we do is that we take samples from different microbiomes of specific environmental places where the conditions are very extreme. Say you have hot springs, or the salt content is very high, or there is a metal this thing contamination. So the microbiome, the microbes that are living in those conditions, will have a lot of genetic uh, modifications, or differences compared to the normal population, which gives them the ability to thrive in those harsh conditions. So which can also which also gives them the capability. So the organisms living in hot springs, so they will have to code for some proteins that are stable in such a high temperature, whereas we know that all the proteins at high temperature get denatured. So basically we'll be able to look for new enzymes and antibiotics as well. So antibiotics are being coded from different genes, or some of the genetic clusters, synthetic biosynthetic clusters, which can give rise to new molecules. So sequencing has made these things possible. So so how does one do these things? Is that when you look into the metagenomic workflow, so whenever you collect any sample, what you can basically do is you can store it immediately at lower temperatures, and then once you get to the lab, you can isolate the DNA by subjecting the sample to physical lysis or enzymatic lysis, depending on so this needs to be based on the material of what you have taken. uh, and then once the DNA extraction is done, the basic sequencing steps will be used, like library preparation and sequencing is done. With once the sequencing is done, you can go through the quality control of the data, and then with bioinformatics approaches, you will be able to look for the microbes that are present in these particular sites. So when it comes to microbiome, so one of the fields that is being that is gaining a lot of recognition these days is the gut microbiome, and how it is involved in health and disease of uh, the host. So a lot of studies have come up showing that in many of the metabolic disorders like obesity, malnutrition, cardiometabolic diseases, diabetes, that there's a dysbiosis of microbes, where some of the harmful microbes are increasing, and this change is can be uh, a result of the disorder, or it can be the cause of it. So we don't know, but the thing is that, so if we can tip this balance, maybe we'll be able to um, correct these particular disorders and come up with a non-invasive and easier solution to these particular disease conditions. And this has been very medically done. It is already being employed in many of the IBD diseases, where diarrhea, Crohn's disease, uh, where C. difficile infections, where fecal microbiome transplantation is one of the methods, one of the most effective methods used in these particular diseases. And nowadays, with the population studies in place, we are able to see that this particular dysbiosis is also seen in neurological disorders and autoimmune diseases as well. So and colorectal cancer and some of the allergic symptoms as well. So this particular uh, pattern of the gut microbiome can be seen throughout the world in different diseases. So here I'm just taking an example of IBD and type 2 diabetes. So the dysbiosis, what you can see here is that in type 2 diabetes, you can see most of there's an increase in E. coli and Rhomboidia, and then the Oscillobacter in many of the populations around the world. And when it comes to IBD, there are some of the organisms which are increasing in their proportion, and these are able to tip the how the host reacts to any drugs as well. So what pivotal role microbiome plays is that not all of the things that are absorbed by the body are ready for absorption. So what happens is that once digestion occurs, the gut microbiome, the microbial enzymes, they conjugate in them in such a way that they will be ready for absorption. Most of the vitamins that are made the precursor of vitamins that are absorbed by the body are conjugated by gut microbiome. So when you look into this particular part, so there have been quite a few studies, and they have determined like what is the effect of the presence of different microbes when it comes to any medication. Are they hindering it, or are they increasing the efficacy, or are they increasing the toxicity of the particular medication? So so for example, when it comes to type 2 diabetes, so metformin is a very commonly advised drug, and you can see that the organisms Alencella and Cetrella. So these are the organisms which can increase the toxicity of these particular drugs. So knowing what is the microbiome that you have, and what kind of drugs you are receptive to can also play a very important role in treatment of any particular disease. So I'm telling you a task that we are doing in our lab is that, and it is a part of my PhD also, is that we are trying to, as part of the Phenome India project, we are able to collect different samples, stool samples from different parts of India, and we have collected thousands of samples, and along with a lot of diet, lifestyle, and blood and physiological data, along with the diet frequency questionnaires, which through which we can analyze the data and come up with population level patterns, signatures of the gut microbiome when it comes to health and diseases. And as part of this particular study, we also gave individual reports for the participants by making it into a pipeline. So why do these population studies, right? So one thing that stands out with the metagenomics is that you are not just looking for any microbes that are present in your database or you are able to culture. You are able to get the DNA of all the organisms that are present, which may be culturable and which may not be culturable, and most of them go uncultured when it comes to the lab conditions. because of the because we are not able to create the similar environment that is present in their original niche. What metagenomics helps us with that is that whenever we extract DNA, we are extracting the DNA of all the organisms in the community. And when we do shotgun sequencing of these particular genomes, we will be getting reads from all the genomes, and these particular reads can be assembled into genomes based on their characteristics like coverage. So how many times a particular sequence has been sequenced, and the sequence characteristics like tetranucleotide frequency. So what is tetranucleotide frequency? So to give you a very simple example, so you must have heard of GC content. GC content of one particular bacteria, say 60%, some have 50%. So it is specific to those particular species or genera that they will have one particular GC content in their genome. So what part of their genome is is the guanine and cytosine. So in similar way, when you take a combination of 4 different nucleotides, it creates around a combination of 256 different combinations. So the ratio of these particular combinations will be same for one particular species. So you need to calculate the tetranucleotide frequency for all these sequences which you will be able to assemble through coverage. And then what you can do is you can put these contigs based on these characteristic, them you can name them into different bins that they belong to this particular genome, and then you can bin them into different genomes. And these particular genomes, when they are made, you can check whether they are in the known reference database or not. If it is not present, that means you are looking at a novel genome which has not been cultured or not been seen by anyone. So when we are doing this gut microbiome study, we are able to see that quite a few lot of species uh, you know um, lot of species are very specific to Indian population, and that they have not even been reported till now in the anywhere, and they are specific to our own population, and they play a very important role in our health as well. So in order to know this, we need to sequence and get their genomes also. So this is the present tree of life of bacteria and archaeal genomes, where you can see that. So the bacteria are being they are divided into different phylums called proteobacteria, actinobacteria, and firmicutes and such. And these particular bacteria, and even today, when we sequence metagenomes, we are able to find new species that span this whole tree. So genomics gives us that edge over any other culturing or any other technologies. And archaea, especially since they are at the border of this particular evolutionary tree, where there is a transition between bacteria and eukaryotes. So they show us how these small simpler organisms evolved into complex organisms. In terms of bacteria, so what you will be able to when you look into the bacterial cultures from these genomes, what you will be able to see is that you can even check a single nucleotide changes that are present in these strains, and these can change the phenotypes. So like a study in Nature recently came, which showed that the strain level, the SNVs that are present in bacteria are changing with the obesity of the host. So they are also determining how the body reacts to how it metabolizes different or food items as well. So when it comes to the clinical setting, how NGS is used. So say I have a patient with TB or some other infection where we are treating them for months and we are not able to see any difference even after antibiotic susceptibility tests and all those things. So what you can do is that one can take the sample, isolate their DNA and then sequence them. And once the sequencing is done, what either we can what we can do is that we can remove the host sequences, because we already know the human sequence, and we can remove them and we can only look for the microbial reads and determine say in terms of TB infection what this particular strain of TB is resistant to. So you can find out what are the resistance genes that are present, and you can treat them accordingly to get rid of the infection. And when it comes to the prenatal care, what one can do is without any invasive methods, you can isolate the DNA from the maternal blood, and there is a certain basal level of release of some DNA from the baby into the mother's blood. So this is called the cell-free DNA. And this DNA, when sequenced, what you can do is you can check for the what are the different fractions of particular uh, sequences that are coming from different chromosomes, and you can, based on that, you can check if there is a trisomy or triploidy, or there is any changes in the sex chromosomes and these things. So this can prove very instrumental, and this is already being done. It's just that it needs the cost, and the analysis needs to be streamlined so that everyone can have access to it. And the rare diseases, so in case of rare diseases, a different analysis approach is needed, because when you look at a population level data, what you are bound to do is that you will be ignoring the outliers. So which are these rare cases? So when you look at a population, you won't be looking into the outliers too much, because you are more focused on the what is happening in the population. So these outliers are actually sometimes maybe giving rise to rare diseases. Specifically looking for these rare diseases and studying them, and how their uh, how the changes in their DNA are being translated and into abnormal proteins, and how we can make up for it through different analysis. So if we can compile all the genetic RNA and the protein analysis, uh, and you can conclude diagnosis, then you can go into functional studies in vitro and in vivo to start the therapies. So one such example is the case of sickle cell anemia, where that particular mutation can be determined and it can be corrected through CRISPR-Cas systems. A similar work is being carried out in our institute by one particular lab. So these can be the applications that can give us a very treatable solution to the diseases that we are facing. So a recent development in the genomics is also the single cell genomics. Is that what you can do is that so before sequencing, what you will be doing is you'll be sorting the cells. Say if you are able to take a particular section of a tissue, and then you are taking different cells together. So in a normal scenario, I would directly isolate DNA from the whole of these cells. But now what I would do is I would sort them using facts. So based on their morphology or their size, uh, or some kind of dye that I'm able to put, I'll sort these particular cells into different types. So now I know that these particular cells are of single type, and when I sequence these particular samples separately, I'll be able to see I will be able to see what is the difference between these cell types. So this will give us the different expression levels that are present in these cells, and we'll be able to study them differently. So why is such a thing required? So say you have mm, say in case of cancer, when you take uh, so you will be able to properly differentiate between the cancerous cells, tumor cells, and the normal cells of the body, and how with the microenvironment around the tumor, how their expression profiles are being changed, and how they are reacting to the drugs or the treatment that you are giving as well. So one more application of this single cell technology is that you will be able to check for the spatial arrangement or spatial distribution of these gene expressions as well. So when you take a particular tissue, what you can do is these are the grids which are designed. So you can put when you put the tissue onto it, so it will have these particular so it will get little bit pieces of these sample, and based on how the region of this particular sample from one side to the other, you will be able to so you will sequence each of these wells as a separate single sample, and you will be able to see how there is a difference between the changes in the series of these wells, and you will be able to, by this you will be able to see in which direction of a particular tissue uh, the gene expressions are of any particular gene are being changed. And all these things, we might say it very lightly, but need a very high end data analysis part, from which you will be able to make sense of all this data produced. So when it comes to genomics at global scale, so a very live example was seen during COVID. So when we sequence a particular sequence in our lab, we may not have much idea of it in the whole bigger in a bigger picture. But when enough people do it around the world, you will be able to establish a population baseline where you can track these particular changes. So whether it comes to any microbe, or in case of humans as well, so you will be able to track the changes that are happening in the populations. And if there is any evolution in these infections, say the COVID was changing strains every now and then, and it was becoming more and more virulent in the next cycle. So you will be able to track these, and if you are doing it at enough places, you will be able to see which particular strain is being prevalent in which part of the world. And based on this particular changes that have been taking place, you can even track back the where it started from as well. So when you look at these global trends, you will be able to come up with a proper vaccination, or the drugs that can be used effectively used against these particular strains, without having to go for trial and error method again and again. So this will increase uh, the treatment efficiency and the time required to determine the right treatment. So there are a lot of challenges in the NGS as of today, and most of them actually have to do with the cost of sequencing, and how the data is stored and analyzed. So when you actually start with any experiment, you need to know exactly what you are expecting out of the experiment, so that you can streamline and decide a particular method without having to go through the fact that after the completion of experiment, you are not able to find what you were actually looking for. um, and the determination of the relevant covariates. So say you need to have enough understanding of the experiments in such a way that some of the things that just change with the samples are not having an effect on, a bias in your results. And the sample collection itself, when done at population scale, is a tedious process, and carrying it out is also an issue. So the processing needs to streamline and increase. And the data generation, so when you generate the data through NGS, so you are getting in hundreds of TBs. So the kind of data you cannot just store it on any laptop or something, or you cannot process it with normal computational powers. So it needs high-end computational power that you will be with which we can process it. And the need for reliable and reproducible methodologies. So the whatever sequencing technologies and the QC methods and the processing methods that we use need to be streamlined, and so that it will so that everyone who does these experiments will get the same results. So so that the results will be comparable, instead of just giving up on the fact that, oh, our methods are different, we can't compare. And the need for the standard databases that we can use as a reference for different populations, or for the entire world, based on the specificity of the experiment. And the data products, so data products of NGS can be very misled, especially nowadays with the AI slop that has come into play. So many companies can be claiming things that are not even possible with NGS. But the non-general population would have no idea of what is true and what is not. I think recently there was a case in US where they were able to manipulate data of a lot of people, where they claimed that will be able to find your brothers, sisters, and lost families. But what they basically were doing is that they were collecting the samples and fabricating the data. So that was a big scandal. So the reliability of the data and how it is produced and how it is being processed needs to be streamlined, and people are working to make policies and make rules and regulations for such things. So uh, so the data that is being generated here will help us a lot in how we perceive healthcare pipelines. So in the conventional treatment of say in case of cancer, so what we were doing till now, or still doing, is that we are finding out that one particular drug is able to give some effect against cancer, and then we are treating all of the patients equally with that particular drug. And what we are able to see is some people are able to get, some are cured, some are not. So we say that, okay, with for them it was not effective, for some it was effective. But when in fact, if we can go into a little deeper and dig deeper to genetic profiling, and if you stratify the patients accordingly based on their how they are reacting to these particular any of the treatments, you will be able to understand that they have a different tolerance for different drugs, and the treatments can be optimized to a personal level, where you can treat every patients with uh, different treatment strategies, so that the treatment is effective. And this comes so this particular process, data driven treatment, if done in a very proper manner, it can lead us to precision medicine, where uh, there will be different entities involved with doctors, medicine, and the analysis, and the samples being processed. And there ethics and data storage, privacy. So such things come into play, and it it needs support from different domains to make it possible. So the data that can be generated from the omics data of genomics, transcriptomics, um, microbiome, and how all these omic studies can be analyzed properly to get sense of how each of the population or the individuals they are reacting to different treatments. And if we have enough data and we make a proper analysis of it, what we'll be able to do is that we'll be able to properly characterize that a person having this particular disease and this particular genetic content and a profile of these things, how he can be treated properly. So this particular mapping will help us treat each of the individuals personally in a very precise manner without having to go for trial and error treatments. So the conclusion here is that the sequencing experiments are exploding in use today. So I don't think I have recently come across any biotechnology experiments where sequencing was not used. So all of the experiments use sequencing nowadays, even the simplest of them. And this is evident by the fact that so the sequencing data, the NGS data, is projected to reach around 40 exabytes. So 1 exabyte is around 1 billion GBs. So you can see the amount of data that is being produced, and which will be 40 exabytes by the end of this year. And we are just gaining momentum in NGS. So this data can increase a lot over the time. And what NGS can do is that it is giving us real-time diagnostics even in remote locations and clinical settings with nanopore sequencing technology, and where you can take the devices to different places where it is not convenient, or for the patients to come to the medical settings. And the cost reduction in sequencing has gone down drastically, and it will keep going down. So the first ever human genome that was sequenced, so that the whole process lasted 13 years and it cost around 2.7 billion US dollars. But today, in the present time, we can sequence a genome in less than under $500. And the sequencing technology are picking up, and the efficiency, if goes to the expected level, this can go drastically down. And the rise of AI and ML methods is a very hopeful technology that can support NGS to be a very promising feature. Is that we can use AI interpretations to optimize our pipelines for clinical reporting. And this can help us go through a lot of data at the population level and at the individual level as well. And with all of these combined, NGS can offer the promise of personalized medicine in today's in today's world, where we are struggling to understand the advance of treatments like cancer and other comorbidities like diabetes, obesity, and such things. So NGS can be used in all of these settings to understand what is happening in the disease and health in a much better way, and it can give us to come up with a better diagnosis and treatment plans. So that was it. Thank you. If anybody has any questions, you can ask.
Vikas, it was a very comprehensive presentation, I must say, and very patiently done with that new topic. For the audience, it is relatively a new topic. Thank you for that. And let's see if in the chat box there are any questions.
Yes.
Yeah. Vikash, there are a few questions in the chat
Yes.
box. Are you able to see?
Yeah, I'm able to see. So what I can see is that, okay, sequencing cost is a major limitation for large scale studies and also for advancing personalizing. Are there any new technologies that can reduce these costs at scale? So yes, so the Illumina technology and the nanopore technology, so they are the up and coming ones. So they are trying to reduce the cost by increasing the efficiency of their sequencing technologies, by which you can sequence more and more samples in one particular one in a single run. You can multiplex a lot more and more samples. But yes, we are limited by the technology as of now, but hopefully we'll be able to see a growth in the technology and a reduction in the cost as well.
I think in this cost that you mentioned is for the whole genome sequence, right?
Yeah, yes ma'am, yes, the
There
human
are
genome.
other options such as,
Yeah, definitely
depending on your
GSA,
choices
you
and
can, yeah.
exam, yeah,
Yeah, you can go for just variant callings where you don't need to sequence the entire whole genome. But
yeah,
like I said, like you can target the genes that are you are interested in, and you can only go for those variant calling
yeah.
for your
And there are
application.
array based methods also where only the SNPs are kind of analyzed. I mean,
Can be
then
detected. Yes,
yeah, or there are exomes. So this is the cost of the whole genome sequence that you mentioned, I believe.
yeah,
Depending on
yes,
the
ma'am.
question, one can do different opt for different
Yes,
methods to look at to compare the groups for genetic variants that they may have. Yeah, and the first question was how can metagenome assembled genomes be
be applied
applied
in
to
studying.
human gut microbiota or environmental ecosystems?
Yeah, so metagenome assembled genomes. When you assemble any genomes from one particular microbiome data, so you will be generating the genomes of all the organisms that were previously that are present in the sample, which may be known or unknown to our existing data, our reference databases. So say in our say in I'm just taking in Indian population, or say in any environmental conditions. So what say some of these organisms are not cultured, or they are not known in the reference, and they are giving rise to a particular property of the microbiome, which are very specific to that particular niche, or say causing certain type of diseases in any population. So understanding these uncultured organisms that are specific to any microbiome, say in Indian context or in any other country population, will be very interesting. And in case of environmental ecosystem, like I said, so the environmental conditions are very tough and very different. So the organisms living there will have a very different profile compared to the normal organisms. So that ability of those particular organisms to able to code for a, say, a heat resistant enzyme, which can like the tag polymerase that is heavily used in PCRs, was taken from organisms that was from a hot spring. So things like this, so they can increase the range of the applications that we have today. And um, there are a lot of applications. So in our lab, we are trying to characterize newer CRISPR-Cas systems from these organisms, from these new mags that we are able to generate from yeah, extreme environments. And even in case of human gut microbiome, we are able to see that we can see a very different metabolism of some of these organisms that we have assembled. So NGS is used for spatial sequencing also. Then how we can distinguish the local gene regulation? and system.
Yeah, I can just say in few words that
Yeah,
the
okay.
special gene sequencing is not for understanding the system level response, but it is for the specific diseases such as cancer, which has heterogeneous tissue and microenvironment, and therefore it is very important to study that in which part or of the cancer, what kind of genes are expressed and how they are contributing to different characteristic nature, which is offering it a resistance to drugs, or it is likely to spread. So that kind of a local regulation is uh, important to study there, so that accordingly the treatment strategies can be planned. So different applications have different questions based on which they are applied. As even in some cases where there is a local affected tissue versus non-affected tissue, and which is very adjacent to each other, and then you want to see how they are actually cross-talking in the micro environment which is shared by them. So for that, these kind of things, exactly at what location, what gene regulation is happening, to study that, the special genes, special transcriptome, or that is being used. So yes, different applications of NGS for different questions is what I would say.
Yes, and this becomes a very crucial thing when we need to design our experiments. Otherwise, you'll be spending a lot of resources and money on the wrong experiments, thinking that you will be, getting the results. Okay, I think that's it. Yes.
Yeah, I think there are no more questions, Vikashi.
Yeah, yeah.
So, as already in previous sessions, I told that we have a Google Classroom. So, if there are any material which we can
Yeah,
share
yeah.
with the, yeah.
Yeah, definitely. So I have a list of some of the reference materials that I use for the slides, and which will be useful for the, for understanding the basics also. I'll share them after this
Please,
lecture.
please, please. So, thank you so much, Bhavana madam. Can we close the session now?
Yes.
Yeah. Thank you so much. Thank you so much, Vikas
In
ji.
the classroom, we will be sending few questions also, but if somebody thinks of some new question, they can also post it in the classroom.
Yes, yes, please. So I request the participants to use the classroom platform
Interactive way.
to ask the question and to clarify the doubt and learn more about the translational research and genomic sciences. So thank you so much, madam.
Thank you.
Thank you so much for this session. Thank you so much, Vikas ji, for this wonderful session. Yes.
All right. Thank you.